Hello there, I'm Cecilia Sanders. I am Life Sciences 1A, and this is my BlueSky project. My project blends my interest in the chemical origins of life and the quest for alternative and innovative forms of energy. So it's a form of bioengineering where we take our inspiration from the earliest prebiotic processes, the precursors to metabolism that might have existed at the bottom of the early ocean or at the edge of drying seas, and then take those models and apply them to a better and more effective microbial fuel cell. So using the metabolic processes of simple microbes arranged in a mat to direct current down an electrode, and then that can be coupled to electronic devices, hopefully on a larger scale. Microbial fuel cells are under investigation by a number of groups presently, some of which are looking at microbial mats as a form of sewage treatment, some of them which are looking to turn that sewage treatment into a source of energy if you can generate current from microbes' metabolic processes, which would oxidize those organic materials and then use the resultant electron transfer to power other processes. Currently, some simple, smaller microbial fuel cells are used in studies related to ocean chemistry, simply to power experimental setups at the bottom of the ocean or in laboratories. So there's a variety of processes as long as you can develop an effective energy efficient setup. In terms of whether you could use something like this on a household scale as a source of energy, for the houses or for your dorm or dorm room is way far off. The first question is really interdisciplinary question of how you understand the metabolic processes of the microbes that you're using. Where did these microbes come from? Are they derived? Are they surface dwellers? Are they thermophilic? Are they halophilic? And under what conditions do you have to keep them in order to continue growing, continue these critical oxidation processes? You have to worry about their level of contact with the metal electrodes. We also don't have great constraints on the magnitude of the current that you can generate with these systems. So microbial fuel cells are a very interesting prospect for the distant future on those scales, but for now it's sort of interesting just to think that here's a way that you can utilize biological processes on an industrial level and couple those studies to further investigation of early Earth processes and early Earth chemistry. My proposal sort of blends several stages together. The first of which is investigating extremophilic microbes from either hydrothermal vents at the bottom of the sea. Those are sort of our best candidates now, and looking at how they act as modern analogs for some of the earliest precursors to living organisms who might have grown up at the bottom of maybe a slightly more alkaline and a slightly hotter but similar ocean conditions at the bottom of a primordial sea. So part of the project is understanding these microbes, figuring out is there an appreciable difference between the redox reactions that they propose, and looking at each strain's individual versions of this process we call extracellular electron transfer. Most living organisms drive their metabolisms with this gradient of ions or protons across some kind of membrane. But these organisms and the ancient proto-organisms that they're distantly related to, they're capable of propagating those gradients outside of their own bodies, which is useful for us because we can tap into that electron flow for the purposes of the microbial fuel cell. So first stage of the project is looking at different strains and seeing how effective each of these strains is at this process of extracellular electron transfer. And then the second stage is working through different variables to design a better and more effective microbial fuel cell. So that might involve setting up separate tanks where you're growing these different strains under a certain like normalized set of environmental conditions, changing the shape of the electrodes that you use. So if you're using bacteria that form mats versus biofilms versus filaments, what is the contact like between the man-made metal electrodes that we're using and the microbial colonies in the mat? Another layer of complexity that you could add on top of that is the specific growing conditions for the microbes themselves. What are you feeding them? What are you using as their energy source? So as you test these different, as you look at these different layers, these different strains of bacteria, these different contact surfaces, these different biofilm structures, you can begin to figure out which combinations of which variables might work best to create this microbial fuel cell. And all of this, of course, under the blanket of trying to emulate these prebiotic redox processes. LS1a was this excellent survey of chemistry and biochemistry, and so we got a little bit of a flavor of genetics, a little bit of the flavor of just basic redox chemistry, a little bit of the flavor of complex organic chemistry. And so all of these things sort of would be necessary to run the experiments that I'm talking about. So even though the project that I'm proposing doesn't specifically work with like running gels and like trying to figure out the genetic makeup and genetic history of each individual strain, we're still applying a lot of those principles. So probably the biggest topics from LS1a that influenced my decision for the project were redox pairs, and this idea of ion gradients in the cell, and the idea that you can take the propagation of this voltage, you've got a voltage going across a membrane, and you can take that and couple it to increasingly complicated processes. So in the earliest oceans, that coupling of voltage across a membrane to organic chemistry is what bridged the gap from an inorganic world with just random spontaneous processes and electrical discharges and black smokers at the bottom of the ocean. So our connection from that inorganic world to the earliest sort of biotic world. And so that's a very fascinating transition that science doesn't completely understand yet, and you have so many groups working on so many different facets trying to put together that chemical puzzle. And then you have the modern application for it. What can we learn from our earliest ancestors? So that plays into the genetics of the situation as well, like the fact that the smallest modern microbes to the largest, most complex multicellular organisms still bear the signature of these early simple redox chemistry, and our very, very basic structures. So that was the influence of LS1a on my choice. Okay, so different groups are working to sort of tweak different aspects of these microbial fuel cells, but there are some variables that haven't been as well studied or as well researched. Ultimately, in changing the strain of the bacteria, in messing with the level of contact and the structure of the microbial film, our hope is to get greater current. You want to have if you have more contact between the microbial mat and the electrode itself, that's fewer electrons escaping into solution and more electrons being guided down your electrode and used for whatever electronic processes that you want to drive with it. So the ultimate goal is to maximize the electrical potential that we can get out of these cells. So my hypothesis for each individual parameter of the experiment would be that if you have the typical view of the electrode in the simplest images is you have your you have just two bar electrodes, and then you have a current passing between them, and you'd have like microbes somehow maybe surrounding it in each side of the electrochemical cell in some sort of solution. But I think I predict that you'd be able to maximize that potential if you can increase the contact surface. So maybe you have electrodes that are more like solid electrodes that are more comb-shaped. And maybe you could have filamentous bacteria that would form colonies that would fit between the bars of the comb, or coat them with some kind of film. So that would be the shape and sort of conformation that I would expect to have the most favorable effects on the output and the potential of the microbial fuel cell. But it's quite possible that there are unforeseen effects of having these globs, what you'd essentially be building of microbes together. Maybe if you have more contact with the electrode, you lose contact with the solution, and thus the access of individual microbes inside of the mat to the oxidizable material, their fuel, their food. So those are possible things that could come into play.